Everyone to today's FPWR or today's webinar. Twice a year, we hold our webinar, What's Happening in PWS Research. And today, Dr. Theresa Strong will give you updates on some of FPWR's current initiatives, as well as details of the 12 grants that were recently selected for funding. The work we're going to share with you today has only been made possible by the very generous support of our community. So I would love to thank all of our donors and fundraisers for your incredible dedication to PWS research. We have a lot of material to share with you today, but we will reserve time at the end of Theresa's presentation for questions. So please feel free to drop any questions you may have into the questions panel. And with that, I'm going to hand things over to Theresa so that she can jump right in.
Great. Thank you so much, Susan, and thank you everyone for joining us today. I will go ahead and share my slides and we'll get started. So today I'm going to fill you in on some of the research activities that are going on, as Susan said. We'll talk a lot about the new grants that have just been awarded. So these are grants that were submitted in the spring and are just starting to get underway now. But we will leave time for questions and discussion. So as Susan said, please add your questions to the question and answer box, and we'll make sure we get to those at the end. So just a reminder about the research process, which does not always go in a straight line, but basically from left to right usually starts with discovery research. So in our case for PWS, understanding how PWS cells or animals or people are different from typical cells. Once we've identified some targets or some differences between PWS and typical, we can move to identifying drugs or other interventions that might bring the PWS cells closer to the typical. And we do that in the preclinical development, usually testing those in cell models or animal models of PWS. And then clinical development is the evaluation of those interventions in phase 1, 2, or 3 clinical trials, hopefully eventually leading to an approval of a drug or an other intervention that can then be applied to our loved ones with PWS, so to the population. And that phase of research is really optimizing the therapy, optimizing the care for individuals with PWS, be it a drug therapy or a therapeutic, for example, a behavior therapy, to have that be as good as it can be for our kids. So FPWR really works to accelerate every step of this drug development process and foster collaborations. You'll hear several examples of collaborative work that's going on, in the PWS field. So we really support those initiatives and make sure that basic scientists are talking to clinicians and vice versa. I did want to mention a few recent papers that have come out with FPWR support. So this is projects that we have supported in the recent past that are now coming to fruition and being presented in the medical literature. And you'll notice we seem to have themes with each grant round. And one of the themes in sort of the latter half of 2024, I think, is gene therapy for PWS. We have a couple of grants that are gene therapy based. We've got a lot of activity in the field around gene therapy. So gene therapy is really taking off for a lot of single gene disorders. And then for a more complex disorder like PWS, people are really diving into how one might use gene therapy approaches to treat PWS. And so there are 3 different papers that have come out from 3 different groups using 3 different approaches to address PWS genetic therapy. So the Gershbach group that is using CRISPR-based therapeutics, Younghui Zhang's group that is using small molecules or drugs to do gene therapy or gene activation, and then the Butler group at UT Southwestern using oligonucleotides to also change the epigenetics. So all of those have come out recently. I'll also point you to a couple of other papers that have come out. There was a nice paper by Dr. Lucarelli, who we funded, I believe, about a year ago to look at hospital records for individuals with PWS. We will write a blog on this. It just came out really recently. Looking at what sends people to the hospital with PWS, and this is an insurance claims study, and it really nicely complements our own natural history study, the Path for PWS Natural History Study. So between our study and Dr. Lucarelli's study, I think we can get a real good sense of why individuals with PWS are ending up in the hospital. And then another paper from the Australian group, Lauren Rice and her coworkers, looking at cardiac activity and oxytocin and vasopressin systems, which continue to be a really important biological system in PWS. I also wanted to mention a preprint, so a paper that is available here, and these slides will be available so you can click on it. We also have a blog on our website about this, and it's looking at COVID-19 and PWS. So this is looking at hospital records. Again, this is led by Dr. Lucarelli, looking at hospital records for individuals with PWS who were hospitalized with COVID-19. Now, we had heard some anecdotal stories about COVID-19 being relatively mild in individuals with PWS. And there were some surveys that were nice survey that was done by IPSO that generally showed a mild course. However, in this case, it's looking at all of the hospital records in the US and who was hospitalized for COVID. And it really paints a different picture, and one that shows you that you need multiple sources of data to really understand, parts of PWS. So this compared about 300 individuals who were hospitalized with COVID to the 4 million non-PWS individuals that were hospitalized with COVID. And basically, it showed that individuals with PWS had a much harder time. So they were younger when they were hospitalized, their hospital stays were longer, they were more likely to pass away, and they were more likely to require a ventilator. So this really says that we need to be more cautious with our kids and adults with PWS with respect to COVID. Now, COVID has changed over the ensuing couple of years. We now have vaccines that can make the clinical course milder, but I think the take-home message is to reduce exposure to COVID, make sure vaccinations are up to date so that we can avoid any hospitalizations. And you can, again, there's a blog that goes into more detail on our website. Okay, so I'll move now just to give a very brief recap of our meeting that we had, our symposium that we had in Atlanta at the end of October, which very brave individuals came to, because we also had a hurricane at the same time. Hurricane Helene came right through during the meeting, but many individuals were able to get there anyway, which we were really appreciative of. With respect to the research symposium, we had about 40 oral presentations and 20 poster presentations of the work that was mainly funded by FPWR, but some other work as well. And there were a lot in the way of both basic science, discovery research, as well as clinical research. So everything from cells to, pooping stories and clinical issues in PWS. We were also really excited to give our trainee travel award. So we had 17 individuals, young investigators, graduate students, postdocs, that kind of thing, who are working on PWS that we were able to support in coming to the meeting and presenting their work. And a really important part of our research symposium, again, is the ability for collaborations to form and for people to exchange ideas. So we really try to have a lot of different side meetings and opportunities, less formal than sort of standing up and giving a presentation, so that people can talk to each other and develop their project in the context of the field. So we had a side meeting for the clinical investigators, the PWS CLIC, the Clinical Investigation Collaborative, which is a network of about 30 different clinical sites in the US and Canada, where they could exchange their ideas about how best to care for individuals with PWS. We had the Clinical Trials Consortium, which is academic clinicians, the representatives from all of the companies that are in the PWS space, and then the patient group representatives, so IPSO, PWSA, and FPWR, talking about how to improve clinical trials in PWS. We also had a roundtable about MAGEL2, which is one of the really important genes in the PWS region. And there's a core group of investigators who are working on MAGEL2 who, spend a couple of hours just sharing ideas and sharing what they were doing so that they could, not be repeating each other's work and be building on each other's work. And finally, something that we tried this year that was really well received were breakfast roundtables. So before the meeting, we had, a breakfast table on hyperphagia and a breakfast table on cell models of PWS. And it allowed individuals to get together, talk about their work, meet new people who were interested in what they were interested in. And we also asked for feedback about how FPWR can be supporting each of these areas of investigation. And we literally got pages and pages of feedback, which we are reviewing and will incorporate into our plans moving ahead. So that was a really exciting and productive way to spend the morning. With respect to some of the initiatives that some of our, collaborations are doing, I just wanted to mention one of those, and that is an effort by the CLIC, so the clinical group, to contribute protocols to a new website, RareCap. So RareCap is the clinical protocols for rare disease patient care. And so it's many rare diseases. It's all freely available on the web. It's available to both clinicians and to parents. And we've developed a PWS site within RareCap, and we are putting protocols in so that if you take your child to the ER and that physician is not particularly familiar with PWS, there's a place they can go to understand some of the unique aspects of PWS in a clinical setting. So, for example, the CLIC recently did a study looking at feeding tubes, the use of feeding tubes in babies with PWS, and their conclusions and guide recommendations are available now in RareCap, so that if a baby with PWS is born in a hospital that doesn't have that much experience, they can now go to the RareCap site and see what the experts would recommend. So we're excited about that. It's early days. There's just a few protocols in there, but we'll continue to build that site. We also want to mention that our Schaff-Yang families are, before the end of the year, will have a new registry. So for those of you who are PWS families and not familiar with Schaff-Yang, Schaff-Yang syndrome is caused by mutations in MAGEL2, whereas kids with PWS are missing MAGEL2. And there's a lot of overlap in the clinical issues, but there's also some significant differences between our different populations. So we thought it was really important for the Schaff-Yang families to have their own registry where they could really focus on the things that are most important and most challenging for their kids. So we are currently working with NORD to, get the last bugs. We're in beta testing, and we expect that to launch. So we're really excited about that. Please keep an eye out, and, we'll let you know as soon as that is ready. And the other big news that is on the horizon potentially is DCCR, or diazoxide for Prader-Willi syndrome. Just a reminder that has, as all things in research and clinical development, has not gone as quickly as any of us had hoped, but it does show the value of perseverance. So just as a reminder, the road for DCCR in PWS actually started about 10 years ago now. And FPWR funded a study at Dr. Komonis's site, just a small study to see if DCCR might have any benefit on hyperphagia. And it was from that small study that the initial positive results came out that encouraged a larger study, which was disrupted by COVID. But again, Soleno, to their credit, really, just stuck to it and kept pushing, and developed an additional withdrawal study that would support the positive results of DCCR and PWS. All of those studies were now submitted to the FDA in a new drug application by Soleno in June, and we're now awaiting a decision from the FDA about whether DCCR will be approved for the treatment of PWS, and we expect that before the end of the year. So it's very exciting times. If it gets approved, it would be the first new treatment for PWS since growth hormone, which was about 25 years ago, so a very long time. So that's really exciting and has the potential to, have a big impact on our kids moving forward. But I would also caution that our work is not done. We know that, we've seen the positive findings from the DCCR studies, but it's not going to be a drug for every person with PWS. And, there might be some room to have additional therapies to complement DCCR if it's approved. And I think that we want to, as a community, make sure we have all of the possible tools in the toolbox to address PWS. I think that's really important, and that we really want to capitalize on this opportunity to, develop as many therapies as we can. So this is just a reminder that there are many active PWS trials that are either active or soon to be active, and I would encourage all of you to consider whether this might be something that you could enroll in. So the Soleno study is currently closed. The NDA is submitted, and we'll know the answer soon. I will mention there are a couple that are enrolling. I think I have it on the next slide. So if you think about, what is important to move ahead with PWS, I think with respect to the open clinical trials, you can think about, well, what is the challenge that I'm facing? So FPWR is sponsoring, for example, a study of vagus nerve stimulation in PWS, and it's specifically targeting those kids and adults who have severe temper outbursts, or a lot of temper outbursts, or temper outbursts that are interfering with, somebody's daily life. And this is a device trial, which is maybe attractive to some people because it's not a drug. We're currently enrolling individuals aged 10 to 40 at sites across the US. Acadia is looking at carbetocin, or that's the drug that showed some promise in Levo's phase 3 trial, but was not yet approved by the FDA. So Acadia is currently enrolling people at the dose that was effective in the Levo trial to gain additional evidence, hopefully, that there is improvements both in hyperphagia and potentially in some of the anxious behavior that we see in PWS so often. So, that's another option for individuals with PWS. If your child has difficulty with daytime sleepiness, Harmony Biosciences is evaluating pitolisant for daytime sleepiness in PWS, and there are several sites that are open for that. Aardvark Therapeutics has completed a phase 2 study, and based on encouraging results there, they'll be opening a phase 3 study at the beginning of 2025 in the US and outside of the US, evaluating their gut-restricted drug and its effects on hyperphagia. So all of those are, depending on whether your family struggles with hyperphagia, whether they struggle with anxiety, whether the struggle is with temper outbursts, you might want to consider any of these trials. And I'll also give one final shout out to a pilot study that is being done at Harvard University, or Mass General, Brigham and Women's at Mass General. And that is a device as well, transcranial magnetic stimulation. And they just need a few families to complete that study. And it's best suited for someone who is in the New England area because it requires multiple trips, but it's a really interesting approach based on some brain imaging findings that this group developed in collaboration with a group from University of Pennsylvania. And it's really trying to stimulate some areas of the brain that they showed to be important in hyperphagia in PWS. So if you are in the New England area, I would ask that you consider enrolling in that study, and that study is for adults with PWS. You can learn more about all of these studies on our website and look at the inclusion criteria and see if your loved one with PWS is suitable for those studies. Okay. So now we'll go through the new grants that we have funded. So we were very fortunate. We had a really strong group of grants that came in. It's clear that many researchers are now very familiar with FPWR and are, building programs around PWS research. So we have awarded 12 new grants, about close to $1.3 million, and we'll go through those. As I said, each grant cycle tends to have its own flavor, and this grant cycle, we saw a lot of basic science research and a lot of gene therapy, so we'll go through that, and a lot of testing new potential drugs in models of PWS. Okay, so here's the 12. I won't read them, but we'll go through them by category. So we'll start with the very basic science, the genetics, and cell biology. And there were 4 projects in this category, and they're really, several of them are based on looking more closely at the function of some of the genes in the PWS region. All of the blue genes in this diagram are the PWS region genes, and the ones that seem to have a whole lot of importance are, for example, MAGEL2, which is important in both PWS and Schaff-Yang syndrome, and the SNORDs, these small RNA genes down here, as well as this gene that gets not that much attention, the SNRF-SNRP-N gene. So all of those are going to be genes that we're talking about over the course of the next few grants. So the first project is looking at phenotypic differences or characteristic differences between PWS and Schaff-Yang syndrome. So this group at Heidelberg University, the LASH group, is looking at MAGEL2 specifically, and their goal is to understand the role of MAGEL2 in both PWS and Schaff-Yang syndrome, and they're using some advanced cell imaging techniques and protein analysis. So we're excited about this study because it brings a new investigator into the PWS and Schaff-Yang field. This is someone who is experienced in some of these really advanced cellular techniques of understanding what proteins do in cells. They're using some innovative technology where they're going to tag the MAGEL2 so that they can see what it's doing and where it's going in the cell. And they're generating these cells that can then be used by other researchers around the world to ask their experimental questions. So the long-term goal of this project is to understand the normal role of MAGEL2 so they can better understand what happens when it's mutated, as it is in Schaff-Yang syndrome, or missing, as it is in PWS. And again, understanding at the cellular level, what are the changes that are either unique to Schaff-Yang syndrome, unique to PWS, or shared between the two. The second project is this study from Dr. McManus at Carnegie Mellon University. And he is looking at, he's again, a relatively new investigator to the PWS field, although he's collaborating with Rob Nichols, who is very experienced in PWS. And he is looking at this one gene in the region, this SNRF-SNRP-N gene, which is a very unusual gene. If there is anything that characterizes the PWS region genes, it's that they're all very unusual. So SNRF-SNRP-N is unusual in that it is one gene that makes 2 proteins, and that's very unusual in mammals. It's not unusual if you're a bacteria, but it is pretty unusual in humans. So he'll be looking at what is that process, and then what is the role of each of these proteins, the SNRF and the SNRP-N. These are right smack in the middle of the PWS region and likely have some important roles in typical cells and roles that are missing when we have PWS. So we're excited about this project because it's a collaborative effort from an RNA splicing expert, that's Dr. McManus, and an experienced PWS researcher, that's Dr. Nichols. Understanding the role of the SNRP-SNRP-N proteins in PWS is really an understudied area. And so we're excited that they'll be bringing their expertise to this question. Again, this is a basic science project, but we hope to improve the understanding of some of the underlying basis of some of the neurobehavior and the endocrine problems that we see in PWS cells when we lose expression of SNRP-SNRP-N. The next project is one from Dr. Elena Buchkova, who is looking at the genes outside the PWS region and how they might impact the severity of PWS. And they're using a data set that was developed with support from Soleno Therapeutics. So all of the individuals who enrolled in the Soleno trial had a DNA analysis done, and that coupled with some of the clinical characteristics like weight and height and blood pressure and some other characteristics allows Dr. Buchkova, who studies large gene sets and in populations, to understand how some of the genes outside the PWS region might be influencing the severity of PWS. And this project really complements a study that we're doing at FPWR, looking at whole genome sequence analysis of individuals with PWS for the same purpose, but using a different technique. And this study will allow Dr. Buchkova to collaborate and us to pool all of the data and strengthen the analysis of those genes outside the PWS region that impact PWS severity. So we're excited about this. This is a second year of funding for this project, and there was excellent progress in year one. It's combining genomic data from the DCCR study and the PWS Genome Project. This is a great collaboration, and I think that the expertise of so Dr. Worthy at UAB has done the whole genome sequence analysis. Combining their brains, I think, will be a great thing. And the ultimate goal of this study is to understand the individual risk for different symptoms of PWS. So we know some individuals with PWS, for example, have seizures, but most don't. And some individuals have bad skin picking, but others don't. And are there risk genes outside the PWS region that may be influencing that? And the other thing that it may do is by understanding those genes that are influencing the severity of PWS, it may give us some new targets to try to modulate those impacts. And I think this is the final in this group is a second year of funding as well for Dr. Friedman at the Rockefeller University. So Dr. Friedman is a big gun in the obesity field. So his group discovered leptin probably 20-some years ago now, which is the hormone that regulates fat mass in an individual. So he has a long history of working in the obesity field and using molecular techniques to understand the basis of obesity. And he has been working in the PWS field now for about a year and a half. Looking at different subsets of neurons in the hypothalamus. So the hypothalamus has different classes of neurons, some of which may be more important in driving hyperphagia. And his group has identified a particular subset, these LEF1 neurons, and a novel gene, NOVA1, that might be driving the hyperphagia. So their initial studies have identified these possibilities, and now we'll be funding him to look at that further. So they're going to be looking at the function of LEF1 expressing neurons in feeding behavior and energy balance using a PWS mouse model, and they'll determine if targeting this gene NOVA1 improves weight control in this PWS model. So we're excited about this project because it is a very experienced researcher in the field of obesity, really understands obesity very well. They've made excellent progress in their first year, and they've identified these neurons and this gene that may be important in PWS, and they're using a lot of innovative methods to advance their research program. The long-term goal of the study is to identify drug targets specifically for the LEF1 neurons and for the NOVA1 gene and evaluate their potential as a treatment for PWS. Okay, we'll move now to the projects that are targeting new targets for therapy and evaluating those in different models of PWS. And you see 4 of them here. The first one is from an experienced PWS researcher who has been studying these SNO RNAs, or the SNORDs, for a very long time, and did some of the seminal work showing what these small RNAs do. And their group has now developed a new mouse model of PWS that is missing SNORD 115 and the SNORD 116 genes. And they've characterized that and shown that it has some of the early characteristics of PWS. So they're born small and they have that failure to thrive that is characteristic of babies with PWS. And what they'll be doing in this study is testing some interventions. So their question is this: if you intervene and are able to sort of overcome that failure to thrive and those early characteristics or phenotypes of PWS, does it improve the long-term outlook? So does it change energy balance and eating behaviors later in life? And so they'll be testing interventions in mice and then looking at the impact later in life. So we're excited about this project because it is another excellent collaboration between Dr. Cavalier, who is at the Medical Institute. Well, everybody's in France, so they're all in France, and Dr. Bourret, who is on FPWR Scientific Advisory Board, and others who are all working who have expertise both in these particular genes, these SNO RNAs, as well as in neural circuitry and that sort of early development of the brain. They're going to be using this new mouse model, and their ultimate goal is to understand or examine the effectiveness of different interventions on those early PWS phenotypes or characteristics and study the long-term effects of these interventions on energy balance in adulthood. The next study is from Dr. John Creemers, who is looking at, or has been looking at, a different genetic disorder that looks very similar to PWS. So this is CMS22. Deficiencies in this gene lead to characteristics that look clinically a lot like PWS. So individuals are born hypotonic and with difficulties feeding, and they go on to have childhood obesity. So Dr. Creemers was intrigued by the parallels between this disorder and PWS, and has been looking at this protein PREPL that is deficient in CMS22 and seeing if that is related to PWS in any way. And the group's previous research has shown that there are mitochondrial defects in PWS skin cells, so skin cells from patients with PWS, and that if you overexpress this protein PREPL, that you can get recovery of some of that mitochondrial function. Again, kind of linking the CMS22 to PWS on a molecular level. So the goal of this study is to, proof of concept, see if using gene therapy to increase PREPL expression in a PWS mouse model will improve the characteristics of that mouse model. So this is an interesting study because it is a potential link between PWS and a different genetic disorder that shares common phenotypic characteristics to PWS. It's a researcher who is experienced, who knows both PWS, CMS22, and other childhood obesity disorders, and it evaluates a potential new therapeutic pathway. And the long-term goal, if successful, it suggests that new treatments for PWS might be aimed at increasing the amount of PrEPL in cells. The next study looking at potential interventions in cell models of PWS comes from Rob Nichols, and this is using these proteins called chaperones in PWS cells. So Dr. Nichols has done a lot of work in the PWS field, and a lot of his work has focused on PWS mouse models and the early characteristics. So right after being born, mice with PWS have a hard time regulating their sugar, which is similar to humans with PWS. And some of the mouse models will die because of that lack of ability to regulate sugar levels and other hormones. So Dr. Nichols has been looking at the molecular level, what is going on in these cells and what are the deficiencies compared to typical cells. And he has found that these chaperone proteins, which help hormones fold the right way or get processed and get matured the right way, don't seem to be working as well in PWS cells. And so this study will look at drugs that are known to activate those chaperone proteins and see if they can improve the folding and maturation of hormones in PWS cells and eventually in mouse models of PWS. So we're excited about this study because it's an experienced PWS researcher. Dr. Nichols has been studying PWS for about 30 years and has, defined many of the molecular changes in PWS genes and cells. We know that low blood sugar is something that is common both in the mouse models of PWS and in humans with PWS, and it suggests a shared mechanism that can be studied in the mouse models. And the hope again with this project, like the previous project, is that if you correct these metabolic problems early in life, it may have beneficial impacts later on in life with respect to energy balance and regulation of feeding. So the ultimate goal of this project is to determine if these ER chaperone proteins, if modulating them, may be a novel treatment for PWS. So if you can modulate the ER chaperone proteins to fold the hormones better, will that have a positive impact therapeutically? And then excuse me, a final study in this group comes from Dr. Ryder's lab. And Dr. Ryder, you may remember, has been studying baby teeth from individuals with PWS. So you can send Dr. Ryder's lab your child's baby teeth when they fall out, and he can pull stem cells out and then make neurons in a dish from them. So it's a very easy way to make neurons in a dish. And Dr. Ryder has been characterizing those cells now in a few years past and has found that they have circadian rhythm defects, which is not a surprise for people who live with Chafe-Yang syndrome or Prader-Willi syndrome. We often see circadian rhythm deficits. And he's found some differences in the timing of how the neurons mature. That may be important in the developing brain of individuals with PWS. And so in this study, he's going to be trying to rescue those characteristics in these PWS cells. And we're excited about this study because he's an experienced PWS researcher. He has these unique cell lines that he's been characterizing. It builds on the work that he's done to date and these well-defined cellular changes in PWS cells. The long-term goal is to lay the foundation for gene therapies where we can see if we put these genes back into these cells, will it correct these specific deficits and, support the development of them as a therapy. Okay, the final group of grants that we funded this time are really looking at sort of more cutting edge therapeutic approaches. So they're kind of the newer techniques, and there's 4 that I'll discuss. The first one is not really a wet lab project, but rather using artificial intelligence and large language models to understand the existing data sets. So we have many different transcriptomic data sets that have been generated by all of the projects that have been funded in the past years. Those are publicly available. And so Dr. Singh is going to be looking at those and applying artificial intelligence to get new information about pathways that might be disrupted in PWS and possible targets for new drug therapy. So we're excited because, obviously artificial intelligence and these large language models are, really important new tools to be understanding different genetic disorders. And Dr. Singh has a lot of expertise in this area. He'll be collaborating with some PWS experts so he can learn about the field and understand these data sets and apply his knowledge to pulling out the best information from these data sets. This is obviously a really fast-growing space, so we're excited to sort of be hooked up with someone who understands all of the possibilities in the artificial intelligence space. The long-term goal of this project is to use artificial intelligence to find potential drug targets for PWS. So basically identify existing drugs that we didn't know might have applicability to PWS, but potentially have applicability to PWS. Next, we have a couple of gene therapy projects. The first is from the Gershbach lab at Duke University. Again, we have a couple of Duke University studies here. Dr. Gershbach has been one of the leaders in the field of developing CRISPR technologies for all sorts of genetic disorders. We're really fortunate to have his group also looking at PWS. They have already shown that they can use some of their CRISPR molecules to activate the silenced maternal genes in PWS cells. And they did that with funding from FPWR. And now they're going to be looking at, okay, so we can activate those genes, but you're probably going to have to keep them active for the entire lifetime of an individual. So how do we make sure that those genes are efficiently and stably activated? So we're excited about this project because Dr. Gershbach is a pioneer in CRISPR-based gene therapy approaches. They've made some fantastic progress in their first year of funding, looking at some of the different CRISPRs that can do gene activation. They've identified sort of where you have to have those sit down to make the PWS region active. And it's an exciting epigenetic gene therapy strategy for PWS. The long-term goal of this study is to develop a one-time transient CRISPR-based epigenetic therapy that will turn those genes on and leave them on for the lifetime of an individual. Another gene therapy approach comes from Dr. Liu at Yale University, who collaborates with Dr. Yonghui Zhang, who has a long history of looking at gene therapy in PWS. So this group has they've actually developed another new mouse model of PWS that has a very large deletion that is similar to the typical large deletion in humans with PWS. And they're going to be applying their gene therapy approach, which is this new non-viral approach called STEP-R. Well, I'm not going to go through the whole thing. You can read it there. This STEP gene therapy approach to the PWS region and evaluate it in this mouse model of PWS. So they've got this new gene therapy method that is non-viral delivery of the CRISPR molecules and targeting the PWS region. So we're excited about this. It is a different. It has differences compared to the Duke University's efforts. It's using a different gene delivery method. It's using a new model of PWS, a new mouse model of PWS, and they have interesting preliminary results. So their goal is to use this STEP RNP DCAS9 mediated epigenome editing as a therapeutic strategy for PWS. And I think finally, we have Dr. Sempleni at University of Nebraska, who is using these new kinds of gene delivery vesicles called milk exosomes and he's going to be evaluating the ability of that gene delivery method to restore MAGEL2 expression in the brains of a PWS mouse model. So the focus of his lab is really making these gene delivery vesicles really specific for their target. So in PWS, we believe that the genes have to get into the hypothalamus in the brain, and that's a difficult task to do. So it's, targeting the genes to the right place in the brain is going to be really important for any successful gene therapy. So this group has developed these milk exosomes, which are non-viral, so induce less of an immune response. And then they are doing molecular manipulations to those vesicles so that they target more specifically to the hypothalamus. So they'll be using these optimized designer milk exosomes to achieve maximal efficiency, delivering genes to the hypothalamus in a PWS mouse model. So we're excited about this study because Dr. Zempleni is a world leader in the study of using these kinds of exosomes for gene delivery. This is an innovative gene therapy approach that has the potential to be highly translatable because you can make these relatively easily and purify them, and they are, probably relatively safe compared to viral-based vectors, which can be safe also, but have some immune response challenges that they have to overcome. So the long-term goal of this study is to create these designer milk exosomes and target the hypothalamus and see if we can increase the efficiency of getting genes to the hypothalamus. So that is all of the 12 new projects that we're funding. As you'll notice, again, a lot of good basic biology, testing of new therapies, development of gene therapy approaches. So I think it's a really strong group of grants, a lot of excellent investigators. We were really fortunate to have some of the world leaders in obesity research, CRISPR technology, exosome technology, studying PWS. So we're very excited about that. Just a final word about the months ahead and the things that we're looking forward to. The global PWS registry is going to get a facelift in early 2025. We're going to move on to a new platform that will give us additional versatility. Before that, we're probably going to ask you to fill out some new surveys around behavior. But if you would, please keep an eye out for those opportunities on the registry. The registry has been so important in advancing our understanding of PWS. It's only because of you and the fact that we get hundreds of responses that we can feel really comfortable about the conclusions that we reach. So thank you, everyone who has contributed to the registry, and please look for additional opportunities. Our Shaft-Yang families, look out, the registry, your registry is coming, and we hope that you'll be able to fill that out just as soon as it's available. Of course, we're all keeping our fingers crossed and waiting for word about FDA's decision on whether DCCR will be approved for use in PWS. We expect that again before the end of the year. But keep in mind that there are still other clinical trial opportunities. Again, we want as deep a toolbox as we can have for our community so that every individual with PWS has options when it comes to therapy. The only way we're going to have that is if our community is willing to consider enrolling in other clinical trials. So please stay informed. Please consider, whether a particular clinical trial is right for you. The Path for PWS study, the natural history study that many of you were enrolled in and did an absolutely fantastic job of, filling out surveys every 6 months for 4 years. We have so much data. It is so informative, and we're pulling all that data. We're working with experts in the field. And so 2025, we're going to be reporting a lot of that back to you. So we have some really great information about, how hyperphagia changes over time and what serious medical events occur and what drugs people are taking for different aspects of PWS, and we'll be reporting all of that back. And then finally, we hope to see you in person and at the end of June for the United in Hope scientific and then family conference. That's going to be in Phoenix, Arizona. It's a collaboration, FPWR with PWSA USA, as well as IPSO. So it's going to be a big shindig of all of the PWS organizations coming together. So we really hope, and hotel rooms are going. So if you are planning on going to that meeting, please go ahead and reserve your hotel room. You can cancel, pretty close to the start of the meeting, and you don't have to actually pay for it until close to the meeting. So you might want to go ahead and reserve that room now. So with that, oh, there it is, the United in Hope Conference. Hope to see you there. With that, I will thank all of our families who participate in research, who support the organization either by fundraising or by providing donations or by filling out their registry records. All of these are really critical to our mission, and we really appreciate you. And Susan, I will take any questions that we have.
Fantastic. So as we've mentioned before, we're open for questions. We're happy to take them. If you have a question that you would like us to respond to, go ahead, pop it into the question panel. Theresa, you talked about 3 projects that are looking at gene therapies, and I thought perhaps you could talk a little bit about gene therapy in general. One of the approaches you were saying would only need to be delivered once, and it sounded like that's unusual. How often does a gene therapy need to be given to an individual for it to be effective?
Well, with most gene therapies, you would like to do it only once, mainly because a lot of the gene therapies that are done with viruses, and we all make good immune responses to viruses, so it's hard to do it a second time. But making sure that a gene stays on for years is a bit of a challenge, and that's a challenge for any kind of gene therapy. In this case, what we're doing is, well, sometimes some of the strategies are delivering genes, so they're delivering like the MAGEL2 gene or the SNORD116 gene. Other strategies are just trying to take the existing gene, the maternal genes that are present in every person with PWS, but are silent, and turn them on. And that turning on of genes is something that is relatively new. And so, what the Gershbach group and others are trying to do is sort through how do you maintain that expression for the life of an individual. And that's really what they are focused on. So, we don't know exactly the time course right now, but we can expect that it is going to be a bit of a challenge to keep those genes turned on over the life of an individual. And so or the Gershbach team is kind of trying to get ahead of that problem.
Mm-hmm. And, when do gene therapies need to be given to someone in order for them to reap the benefits? Can a mature adult take a genetic therapy and have benefits?
Well, that's a great question, and we don't yet know the answer to that. So that's what some of this work in the gene therapy field will be. We'll be looking at the different mouse models, delivering it, early in life versus an adult. I think the lessons that we learn from the real world experience are informative. So for other genetic disorders, it's always better if you can do it right at birth, right? Because then you can incorporate those genes early, and the development, is presumably, more in line with a typical development. But we know now from the early studies in Rett syndrome and in Angelman syndrome that even in adults, it looks like if you do the gene delivery, you may be able to get some recovery of function. And I don't think that was necessarily the expectation when people went into gene therapy for neurodevelopmental disorders, but it seems to be working out that way. Now, it may be different for different neurodevelopmental disorders, so we'll have to see in PWS, but certainly I think that there exists a possibility that it would be beneficial not only for babies with PWS, but for adults with PWS. But it's, again, something that we're just going to have to work through and, we'll get the data on.
Sure. That's refreshing to hear, because my own son is 15, and we still have hopes that he is going to be able to live a very independent and full life with the work that FPWR and others is conducting. How far out do you think that these gene therapies might be?
I mean, that's a really hard question. So I think in parallel, there are a lot of gene therapies that are being developed for other neurodevelopmental disorders. So as I said, one of the hardest things is how do you get these genes up into the brain? And the general field of gene therapy is gaining a lot of experience in that way. So, there are approaches using little snippets of DNA, oligonucleotides, and those are being widely used for Angelman syndrome, for SMA, for other genetic disorders, and the field is gaining experience about how do you make those where they get up into the brain, for example. There are in development for PWS, we have some drug-based approaches which would not face those gene delivery challenges. So that could potentially be something that goes into the clinic faster. Marnie Blewett is using an entirely different approach, modulating one of the genes that controls the epigenetics of the region. And that also may be a faster way into the clinic. So the CRISPR approaches are really cool, but getting CRISPR, which is a big molecule, up the gene up into the brain is probably a bit of a challenge. So all of that is to say, we've got a lot of horses in the race. It's not clear which one is going to be the best one for PWS, so it's difficult to predict a timeline. But the experience in the field of gene therapy, I think, is going to benefit us for any of those approaches. So that's a non-answer, I know, but that's all I can do.
That's okay. That research it's big and hairy, and we know that it's going to take some time. I'm never going to stop asking you how long is it going to take, because that is the question that we all have. We often use the word transformative when we talk about gene therapies. They have the potential to be transformative because they can essentially fix PWS at its very root. But while we're waiting for this transformative treatment, we have, as you've mentioned, lots of other things going on. So earlier you were talking about the timeline for DCCR. It hasn't been fast. We first funded it in 2014, so 10 years later. But I noticed on your timeline, you had the date, December 27th. Let's talk about what that date means or doesn't mean.
Yeah, that's and I'm not an expert in this, but that's the PDUFA date. This new drug application from Soleno, it had a breakthrough designation, which means that the FDA really tries to review it quickly because, it is a new treatment for a disorder that has a tremendous unmet need. And I think the FDA appreciates that. So that is the 6-month point from when Soleno submitted the application. And that is, that's what everybody looks to. My understanding is, if the FDA decided that they needed more information from Soleno and they didn't want to approve until they got that information and were able to review it, they can extend the date. But I think they are gonna, or knock on wood, I think they are working hard to make sure that they have an answer by that December 27th date. So that is set by the day that they submit the new drug application.
Yep. And I can promise to all of you who are here, you will know as soon as we know.
Yeah.
So we're all waiting, less than patiently for the response from the FDA. And knock on wood, it's all the response that we're looking for. So this ends our time. We are right up against the hour, but Theresa, I always appreciate you helping us to digest this information. These grants are not easy to understand. But you make them, I think, about as digestible as possible. So thank you for your work in doing that. We really appreciate it. And for all of you, I hope you have a wonderful rest of your day, and we look forward to hosting you again on our next webinar. Have a wonderful day.
Thank you all.